Hello, everyone, and welcome to episode 2 of the Therapeutic Antibody Series 2026. My name is Amna Jamil, and I'm a webinar and conference producer at Technology Networks, and I'll be your host for today's session. We're delighted to welcome Alexander Hobby, who will be presenting his talk, Detecting and Eliminating Off-Target Binding for Safer Antibody Therapeutics. Alexander Hobby is a strategic account manager at Integral Molecular in Philadelphia, where he works with leading pharmaceutical companies to support large molecular characterization and therapeutic development. After earning his PhD in translational medicine from Temple University and completing postdoctoral research at the University of Colorado, Alex transitioned from academia to industry, driven by a passion for translating cutting-edge science into real-world impact. Today he leads strategic partnerships and supports the adoption of Integral Molecular's membrane proteome array (MPA), helping advance the development of safer, more effective antibody therapeutics. A warm welcome to you, Alex. Following the presentation, we'll also have a Q&A session. We welcome any questions that you may have. Please ask questions using the Q&A system to the right-hand side of the video player. So without further ado, I will now hand over to Alex to begin his presentation.
Great. Thank you. I really appreciate that introduction, and I'm excited to talk to everybody today. Hopefully you all find this interesting. I think the Therapeutic Antibody Series is a great series of talks, and this is kind of in the middle because I think it's important to recognize a piece of the drug development process that sometimes gets skipped over, which is the actual sort of characterization of these antibodies. So I will be speaking today largely about detecting and eliminating off-target binding, specifically using the membrane proteome array for safer antibody therapeutics. So I will just dive right in. I wanted to start by just introducing the company for anybody not familiar with Integral Molecular. We are here in Philadelphia, Pennsylvania. It's actually our 25th anniversary this year, so we're celebrating 25 years of membrane and antibody expertise. And we've worked with many different companies worldwide, but really difficult membrane protein targets and the antibodies to either detect or treat them is our kind of scientific specialty. And that's what a lot of our work revolves around. So we do quite a number of things at Integral Molecular. I'm going to be focusing today on the membrane proteome array, as well as our relatively new CDR scanning service called Paratope Plus. Those are the methods by which we detect and then remove off targets for partners. Both of those fall under the solutions for antibody research and development, but we also do have our own therapeutic and reagent antibody pipeline, as well as a suite of virology reagents and services if anybody is interested in those. If there's any questions about these, I'll answer to the best of my ability, but I can also connect you with the appropriate folks there for follow-up. Wonderful. So I do have kind of an action-packed agenda. I'm going to go through some of these sections a little bit faster than others, but I'm just going to introduce polyspecificity real quick in the beginning, talk about our approach to specificity testing at Integral Molecular, and then talk about addressing the gap in the clinical relevance of off-target binding. Before going into a platform overview, a workflow overview, talk a little bit about the regulatory space, and then end with the story about parallel optimization and removing an off-target from one of our own internal assets. So as I said, action-packed, but I'll be jumping through some of these sections quicker than others. Okay, so I wanted to start with an introduction in polyspecificity for targeted biologics. And we've screened thousands of molecules at this point on the membrane proteome array. We've been in business doing specificity testing for, I think, about 8 years now. And from early lead selection molecules all the way to later stage IND ready molecules, essentially, we actually find that this number 1 in 3 holds true pretty universally across the board. And so 33% of the monoclonal antibodies that we test show some kind of polyspecificity, usually with a single off target, rarely more than 1 or 2 off targets, but you do get the occasional molecule with many. We also know that off-target binding is a major source of side effects and clinical failures for any drug developer. The FDA does require specificity testing prior to clinical studies in humans, and so companies have to do something. And I think one thing I'll go into later in this talk is that the existing methods for doing specificity testing have not always been sufficient. So just to level set with everybody real quick, because sometimes these are used interchangeably, I will be saying off-target and polyspecificity a lot in this talk. When I say that, I'm really referring to CDR-mediated binding to a similar epitope to the target protein. That's as opposed to polyreactivity, which is more of a general stickiness of the molecule, I would say. And it's something, polyreactivity is really something assessed earlier on in the developability stages of antibody characterization. And for some people, depending on how familiar you are with cross-reactivity and off-targets in these biotherapeutics, some people kind of ask us, well, how does this actually happen? Like, do I only need to screen on family members of my target? Is this something that is relatively common? And this is a great example of how even in totally unrelated proteins, you can actually see cross-reactivity. And this is, again, from an internal project of ours from a number of years ago, where we had an SLC2A4 targeted monoclonal antibody. We actually found on the membrane proteome array that it bound this other protein, Notch1, which you can see is extremely dissimilar in both its structure and sequence. So you have kind of a 12 transmembrane transporter that is the actual target, and this off target here is a single transmembrane signaling protein, essentially. And what we found when we actually did some epitope mapping and dug into this a little bit more is that the monoclonal antibody was binding a shared epitope motif, this LGXXGP motif on both of these. And it was actually the physiochemical properties of that binding pocket that was common between the two proteins. And so really this is all to say that a comprehensive sort of screening strategy is really required for appropriately testing specificity for one of these molecules. Screening family members alone is not usually enough to get the full picture of the binding here. And so that's the approach we take, and I'll talk a little bit about that. I'm going to introduce the platform here, but then I'm going to kind of diverge into some other data before going back to it. So I'll just talk briefly about sort of the comprehensive approach that we do take. And as I mentioned earlier, specificity testing has been required by the FDA since a guidance that came out in 1997. And in that guidance, tissue cross-reactivity studies specifically was mentioned as an appropriate method, but they also mentioned that other methods, new methods could be used as they became validated. And so for a long time, for the last few decades, tissue cross-reactivity studies and some other protein array alternatives have been used. And these are essentially today, in our view, relatively outdated. They're not doing the job that we think that they should be doing for specificity testing. And we've been working with the FDA over the last 4 years through this ISTAN program, which is Innovative Science and Technology Approaches for New Drugs, to actually sort of come up with this next generation of specificity testing. And really, to us, the most important aspects of this are that your specificity testing should be actionable, should be all of these things in the bullet points here: biologically relevant, quantitative, quality, and predictive. And for a lot of the assays that have been used previously, they don't meet these standards that are sort of being adopted by regulatory agencies. So just to speak briefly about what it actually is that we do sort of under this umbrella of specificity testing at Integral Molecular, we offer 2 primary libraries for screening. The membrane proteome array is 6,000 plus membrane proteins that are assessed by flow cytometry. So it's a flow cytometry assay where we are expressing in a transient transfection model all of these 6,000 or so membrane proteins and assessing binding, looking at the fluorescent signal off that flow cytometer. For the secreted protein library, which is sort of an add-on library that we have as well, we actually do that by ELISA, so it's extremely sensitive. I'll speak to this in a second. We decided not to take a tethering approach for these molecules, so that's why we didn't do them in a flow cytometry approach. But these are available as an ELISA screen as well. And for both of these platforms, the SPL was launched only in 2024, so it hasn't been around quite as long. But for both of these platforms, the quality and reproducibility of the data is really important to us. And we've done a lot of work recently with the FDA to basically show that over years, lots, operators, over time, the quality of the assay doesn't change. And that's really important for our partners. And then probably most critically, I think all of our library design boils down to how to become or how to be as biologically relevant as possible. I think for the membrane proteome array, the answer there is to do all of our binding in unfixed staining conditions. And that is something that separates us quite a bit from other platforms out there. We're able to assess binding on unaltered epitopes, and I'll show some data later in the talk about how fixation can actually impact binding and epitope expression. And for the secreted protein library, again, these are native protein sequences. They're not tethered to anything. And so this is kind of a broad strokes overview of the different libraries that we screen on when we do specificity testing at Integral Molecular. I just wanted to mention real quick that we are launching a non-human primate library soon. This is actually going to be ready in the next month or so. It's launching with about 250 proteins that are less than 100% homologous from the human library, but this may be of interest to anybody looking for information about preclinical tox species selection, or just looking to make sure that in a lot of their animal studies, certainly in the cyno model, that they're not going to have any unexpected results that could impact the development of that molecule and its movement to the clinic. So just wanted to put that out there. If this is of interest to anybody, I'm happy to speak more about that. Okay, so I'm going to take a step back and talk about the gap here. And I think it's important for us to address the clinical relevance of this, because that's kind of the ultimate goal for all of this, all of these drug development efforts, is getting these molecules into the clinic. And I think the impact of off targets is somewhat sometimes missed on the impact it can have downstream once you've already invested a lot of time and money into these molecules from a clinical development perspective. So I'm going to go through a couple data tables here just to highlight some of the work we've done internally to kind of make this case. We call this the ClinMabs project, or Clinical Mabs project. We've screened actually a couple hundred of these now, but this is summary data from 83 of these molecules. And what we did was take 83 biosimilars from different clinical stage molecules that are in these different phases, as you see here on the table. And of those 83, of all that we screened, we actually found 15 that had off targets that were not disclosed at the time of IND or BLA filing. So as we go down the sort of from left to right here, you can see in the withdrawn category, the rate of off-target, the rate of polyspecificity of these molecules is actually highest. These are withdrawn molecules that were withdrawn for any number of reasons, but they did not make it past the first phase of clinical trials. The phase 2-3 molecules had a slightly lower percentage. Now, I will just acknowledge here that these are relatively low numbers to make any kind of actual statistical inference from, but I think the trend here is what is important, because you can see as we go sort of, as the performance of the molecule improves and we get to these approved on-market drugs, the rate of polyspecificity goes down, and that just speaks a little bit to attrition. But what I want to highlight here is that even in the approved on-market drug category, these are drugs that are in people today, 6 of the 40 molecules that we assessed showed validated reproducible off-target binding that was not detected or disclosed at the time of any kind of regulatory filing. And that's a percentage, again, different from that 33%, because that's all molecules. These are just clinical stage molecules, but that 15% is probably what many would consider to be too high. And certainly that's what we consider to be too high as well. So we dug into these molecules a little bit more. For 35 of the 40 that we screened, we were able to find tissue data, tissue cross-reactivity data, essentially, that was publicly available. So we could actually compare our data to the data that was out there. And what we found here is summarized in this table. For these 7 molecules where the MPA result found an off-target, this is 7 because one of the molecules actually progressed in clinical trials and became approved while we were doing this study. But of those 7, the TCR result was clean. When the MPA result found sort of off-target binding that hadn't been disclosed yet, when we dug in deeper, we found that of those 7, 4 had black box warnings, 2 had severe side effects, and the only molecule that did not show unexpected toxicity, the off-target was actually sequestered to the brain, essentially, and that therapeutic was not actually reaching that off-target to have any kind of effect. So this was really interesting to us. Now, what we're not doing here is we're not able to draw a causative line between the off-target and the clinical effects. That would take a lot more science and experimentation that we have not done. But at the very least, these off-targets should have been detected and understood before going into the clinic, at the very, very least. And when we look at these other two categories here, the sort of next two down in the table, where we found a clean result, the tissue cross-reactivity result was either unexpected cross-reactivity showing some kind of off-target binding or inconclusive. And I think in both of these cases, assuming that the MPA result is a little bit more accurate, this really just represents wasted resources. The inconclusive results of TCR, I think anybody that has done these studies knows that the optimization step for tissue cross-reactivity studies is the bulk of the time and cost associated with those studies. So to get inconclusive results back after all of that can be devastating for a program. Lastly, I did just want to acknowledge as well that the two platforms did agree about half the time. So for 17 of the 35 molecules, both of the platforms agreed here. So I think the gap here is really, specificity testing is required. In theory, companies are doing it, and still there are a pretty large number of molecules with off targets that are kind of falling through the cracks. And that's really what we want to address, is that the conventional methods that have been used for testing specificity in the past are really, what we're finding is that they're just not sufficient for modern drug development tools and modern drug development modalities. So in both these cases, for tissue cross-reactivity studies or these protein array alternatives, they're not assessing native epitope conditions either through freezing or fixation. They are not quantitative. These are really qualitative measurements. They're not validated or qualified by the FDA either, which is technically not a requirement now, but is really important as we see new guidance coming out of the FDA. And I did just want to mention for tissue cross-reactivity studies, I do absolutely acknowledge that it does provide information that a screen like the membrane proteome array cannot provide, which is that tissue level information. So on target off tissue essentially expression would be something that you could get from tissue cross-reactivity studies, but still it doesn't give you the level of granularity and detail that you need to make the informed decisions for the development of a molecule. So now that we kind of have that gap established, I want to just go into an overview of what the MPA and SPL are and how they sort of fill that gap. So we designed our libraries to be as comprehensive as possible. We actually based the design of these libraries off of the 34 human tissues required for tissue cross-reactivity studies. So the FDA has a set of tissues that they've said you need to screen on these 34. We took those 34 and we did an unbiased next-generation sequencing approach to create a global list of membrane proteins from all of those tissues. From that global list, we then narrowed it down to about 94% that we could reasonably and reproducibly express. The 6% that we can't express are either too big, too tricky, or too finicky for really anybody to express. And so they're left out here. But we do cover about 94% of that original list. The library includes GPCRs, ion channels, heterocomplexes, both intracellular and extracellular membrane proteins as well. And then for the secreted protein library, this was actually less of an unbiased method. So the secretome is kind of a moving target depending on how you define secreted proteins in your search terms. And so what we did is we actually curated about 1,200 of the most relevant for drug developers. So a lot of your chemokines, cytokines, metalloproteinases, a lot of proteins that are going to be relevant for your drug in an actual person. That's how that library was really designed. The other thing that's really important to us is being able to screen a wide variety of biotherapeutic modalities. As I mentioned earlier, the technology is advancing at a rapid pace for some of these new modalities, especially in the cell therapy and antibody drug conjugate or ADC space. You'll notice that normal IgGs are not even on here. That's kind of what we screen every single day, but we are able to screen pretty much anything on this slide, as well as some other things. And our motto is kind of, if we can detect it, we can screen it. So we've screened a wide variety of molecules on this array, and the data quality holds up across all of them. Okay, and I did want to harp on this a little bit because I think it's important. I have a little bit of data here to show why screening on unfixed proteins is so important. And the biological relevance of the MPA, again, is a big piece of how we're filling that gap in the absence of that in some of these other platforms. So this is just an example that we generated in our own hands, where we actually had an internal asset targeted to CD19 that we screened on the membrane proteome array. On the left is our unfixed cells. So this is what we always do. We always screen in unfixed conditions. And we did find an off-target that validated that we were able to kind of reproducibly validate for that molecule. Now, we don't typically do this, but at the time, this was an earlier study as we were developing the platform more. And so we ran the equivalent screen, the exact same screen, but in fixed conditions on fixed cells. And we saw actually great binding to the target. The epitope was intact there, but the off-target that we had found in the unfixed screen is essentially buried in the noise. And this off-target would never come up above our thresholds for us to say that this is something that we should evaluate and de-risk as an off-target. So this is effectively a false negative, and this is something that can come from fixation as well as an increase in false positive events in addition to that. So I think fixation is really something that we always try to avoid, and screening on that unfixed array really brings out the biological relevance of the membrane proteome array. One other thing I wanted to mention here is that the quantitation of this information is really important. I mentioned this when I was talking about kind of the next generation of specificity testing. Qualitative metrics of high, medium, low, or tissue expression information doesn't really get you the actionable data you actually need when you're making determinations about the development of these molecules. And so everything we do is built to be as quantitative as possible. So from the primary screens themselves, we're generating binding scores and representing them as what is essentially a confidence interval, but standard deviations from the mean background. From there, everything that we call off of that screen goes into a validation step, which I'll talk a little bit more about later. That is a quantitative 4-point titration where we're looking for actual titrating binding of that off-target and on-target. And then we have a number of follow-on services that we do as well. Our enhanced binding analysis experimental package, where we are literally sort of expanding that titration, calculating EC50s, looking for binding saturation and things like that, so that you can actually see the relative sort of power of that off-target binding, so to speak, as compared to the target. So again, across everything we do, we're really trying to make this as quantitative as possible so that you can actually use the data. And lastly, I just wanted to mention the quality piece, because this is something, again, that kind of, gets taken for granted sometimes. As part of our work with the FDA, we've done a lot of analyses of the reproducibility of the platform and the quality of the platform overall. We've submitted a lot of data to them, and we've shown that over time, again, as I mentioned earlier, the quality of the platform doesn't change. We were ISO 9001 certified just a couple of years ago. We have a lot of controls that we run internally to maintain the library as well. And then experimental and non-experimental controls are factored into every project that we do. So I'll talk about this in the workflow later. Our method is not to just run your molecule, dust our hands off, and give you the data back. Really, what we're trying to do is design the best possible experiment for your molecule, and we can incorporate a number of different positive and negative controls to do that. So again, the quality of the data is extremely important to us as well. Okay, so I'm just going to briefly go into the workflow here, just to give you a sense of how we actually screen and detect off targets at Integral Molecular, because I think this is important to understand. Again, that's the sort of quality behind that. And before I go into that, I just want to mention there are really two times in which we interact with drug developers when they're developing biotherapeutics. The earlier sort of lead selection phase of drug development can be screened on the normal membrane proteome array. This is kind of our standard package where we're screening anything with 3 or more molecules, usually around a dozen molecules or so. And then when drug developers have a later stage candidate that's sort of ready for an IND submission, we have a regulatory ready package, this MPA plus IND package, that has been submitted to the FDA many, many, many times, over 100 times, with great success. So just to mention that we do have these two sort of pieces of the puzzle here for how you can use this array. And when you do a project with us, we're always going to do these 3 phases. And I think the reason this is important is because these phases are not always included in other studies. And it's really important to do all 3 of these because it again, speaks to the quality of the data. So no matter what, we're going to set up your molecule by assessing for the optimal cell line and antibody concentrations. This is in another 4-point titration, looking at background above our negative controls, or signal above our negative controls, I should say. And we do have alternate cell lines that we can pivot to if there's high background in our normal cell line, which is a hex cell line, a human cell line. So that's all that means there. Once we have the optimal conditions, we then move to the actual screen itself, where we'll assess the molecule across the 6,000 membrane proteins or 1,200 secreted proteins in parallel. And then anything that we find on the screen goes through this additional hit validation step for every project. So again, this is something that differentiates us a little bit. This is always included. We're not calling hits off the screen off targets until they've been validated and we've seen binding that titrates across multiple different concentrations. So what we're trying to do here is minimize the number of false positives that we're sending you and minimize the amount of follow-on work that has to be done, because we do recognize that can delay programs. It can delay development in ways that cost a lot of time and money for everybody. And so what you'll see when you get a report from us is that anything that we're calling an off-target binds in a titrating manner at multiple different points in this validation step. And then finally, I mentioned these briefly earlier, but we do a number of follow-on studies, largely whenever we find an off-target for a partner. This is something that we can do just to give additional statistical confidence. We can look at relative binding strength to the actual target. And all of this really goes into de-risking an off-target that we find. And I'll show you a couple case studies later where finding an off-target is actually not the end of the world. We can de-risk them and they can still move forward. Okay, so let me get into the regulatory progress we've made a little bit, just to kind of give you a sense of where we are there, because this is really indicative of a shift, I think, in the field overall. As I mentioned, we've had over 100 IND filings with MPA data. I think after this year, it'll be over 150 or 175 or so. We've never had anybody come back and say that the MPA data was rejected. The only thing we've gotten is questions, essentially, and that was in the earlier stages of this IND package that we have. So for a number of our partners, they've actually submitted IND filings without tissue cross-reactivity data, especially depending on the modality. It may not be required if it's incompatible with that type of immunohistochemical staining. This is one such example from Cabaletta Bio, where they actually found a low-level off-target, de-risked it, submitted without tissue information, and this has been published and is now in clinical trials today. So we're nearing qualification as a drug development tool in capital letters. That's an official designation by the FDA, and I just wanted to speak quickly to the work that we've done to get to this point, essentially. But we are on this last step where we've submitted the full qualification package to the FDA. They are reviewing it. We've gotten some questions back. There's some correspondence between our scientific team and the FDA. And so we expect to get a designation on our drug development tool status within the next, hopefully, 9 months or so. We don't have a lot of insight into the exact timeline, but this quote here from the recent roadmap that was released by the FDA kind of says it all. Once qualified, any sponsor could use the new approach methodology, or the MPA in this case, in an application with the confidence that the FDA will accept the data. So this is essentially going to be a rubber stamp for the MPA to be used in certain cases for specificity testing, for certain modalities, certain therapeutic areas, et cetera. So this is really exciting for us, and we're looking forward to these developments. Okay, a couple of case studies here at the back, and then I'm going to talk about a little bit more about removing some of these off targets. So this is a case study from one of our partners, Embrace Therapeutics, who we work pretty closely with. They had an antibody drug conjugate, or ADC, that they wanted to screen with us that was targeted to this EphA5 or FA5, which is a novel solid tumor target. So it has a high expression in solid tumors. There's very limited expression in normal tissue for this target. And so they sent us a humanized anti-FA5 antibody that they had conjugated to the actual payload, MMAE here in this case. And what they published is essentially looking at all of the family members for FA5. They did run the full MPA screen, but this is published data, so we wanted to use the data that they had in their paper, basically showing binding only to that target and not to any of the other family members here. They did tell us that they used this in their IND filing. They used the full MPA data set in their IND filing with no TCR study, and that IND was approved. This is currently in phase 1/2. You can see the clinical trial identifier there. And they also published this. This graph is from the paper citation at the bottom. So this is showing, especially in the oncology space, you can move very quickly with some of these new targets if you do the membrane proteome array for those filings. Similarly, with the partner Triumvira here, this is for a cell therapy application, but again, similar in sort of scope. Based on the 2024 CAR-T guidance documents from the FDA, we were able to screen just the VHH GFP fusion on our array. That is sort of within the guidelines that the FDA has put forth. So we screened that soluble Claudin 18.2 VHH GFP on the MPA itself. It was a very, very clean molecule. It did not show binding to anything else. And if you're familiar with the Claudin family members, they're all extremely similar. I have another Claudin example for the Paratope Plus example later on. But these are all extremely similar family members. And so showing a clean molecule to Claudin 18.2 without binding to any of the other family members is very important. And similar story here, the FDA granted the IND with no TCR study required. So you can see the publication here with this data, and you can also see the clinical trial identifier if you wanted to look into that as well. And then lastly here, this is the last example that I have for MPA screening. This is actually recently enough that we've had to just de-identify the data as we get permission to use this publicly with the partner sort of on the slide here. But this was a project in which we were sent a T-cell engager therapeutic that had a couple of different targets. The target here, the membrane protein target, was the primary target of the molecule, as well as the CD3 arm of the molecule. And then they screened on both libraries. So in this particular case, the customer elected to do the comprehensive screen and screen on the secreted proteome library as well. We did find unexpected binding to an off-target in the secretome, as you can see here on the right in the SPL graph. But no other unexpected binding to a membrane protein. So the partner here orthogonally confirmed binding to that secreted off target. It was interesting because there was actually potential for this to matter in the clinic. This protein, the secreted protein, played a significant role in the skin. And so there was the potential for dermatitis or adverse events in the skin in the clinic. So they de-risked this essentially with their own animal studies. They did follow-ups to show that it actually would not be a risk. And so they were aware of it, they disclosed it, but they de-risked it. And that really strengthened their entire regulatory submission to the FDA to show that they have actually done the work to do that. So they incorporated the animal data and the MPA data into a weight of evidence regulatory approach for that submission. And this candidate is advancing towards clinical trials in the very near future. Okay, so I've talked already a little bit about immediate de-risking here, and I think what I'd like to end on is a story more about the engineering side of this and the CDR mapping in Paratope Plus. So far I've gone into pretty great detail as to how we can detect off-targets, and this is something that we've more recently gotten into, but optimizing these leads in parallel, including removing or decreasing affinity to an off-target, is something that we can do through this Paratope Plus platform that I'll introduce here in a second. So just to make sure everybody is clear, the paratope is the sequence on the actual antibody responsible for binding the epitope. So the epitope is the antigen side of things. The paratope is the antibody side of things. And so what we're doing when we're engineering the paratope here is we are essentially changing this binding interface. And I'll show you what that looks like on the next slide, or the slide after this one. So this is a multi-parametric challenge, right? I think everybody that has tried to do this in general knows that it does sometimes feel like a game of whack-a-mole. So as you're trying to increase or decrease one factor, you may change another one in parallel. That just makes it very difficult if you're selecting for these things individually. And so parallel optimization here is really the key to the efficiency of this and really the value behind it. And again, about a third of the clinical stage antibodies that we're testing have suboptimal physicochemical profiles. This is a relatively common issue. And I think, again, people that have experience with this certainly would understand the whack-a-mole sort of metaphor here. So what our strategy is, as it says on the bottom of the slide, is really to try to optimize these parameters at the same time and do as many of them as we can at once. So the way that we do that is kind of outlined here, and I have some example data in the next couple slides, but really what we're going to do, we have all the legal documentation in place for you to send us your sequences without any risk to your intellectual property. We do know that those are very sensitive sequences, especially for drugs in development. So you'll submit your sequences to us. We will create a parental construct and we will optimize binding conditions in the assay setup phase, very similar to the membrane proteome array. But then from there, this is where things really diverge. We will create either a Fab or scFv library where we're mutating every amino acid in the CDR region of your antibody to all other 19 variants possible, essentially. And we do that sort of sequentially one at a time so we can generate these large, about 1,000 construct libraries that we can then assess for binding through a high throughput assay afterwards. So the deliverables that you get from this are can be used in many different ways. I think one thing that is really interesting here, which may not be super relevant to this audience, there's the engineering application, certainly, but there's also the intellectual property application or implication of this as well. We recently submitted a patent application for one of our own assets where we essentially patented or are going through with patenting all of the permissive variants that we were able to demonstrate similar binding to with this assay, with this strategy under that same sort of patent umbrella. So we're calling that, we're patenting a genus of those sequences as opposed to an individual species sequence. And so that's something that could be really interesting if anybody's interested in that. I can talk about that, but there's a lot of intellectual property implications for this type of data set. And the reason for that is because the data set is actually quite comprehensive. And it's something where we can show both an increase and decrease or gain or loss of function when it comes to binding either the target or an off target. And we're doing this across the heavy and light chain, all 6 CDRs of a molecule, and we're doing this across all possible residues. So again, we're creating a large number of variants to analyze. This is just to give you a sense of what these data sets look like. I'm not going to go into these numbers really in any meaningful way, but what we do is as we mutate each of these amino acids, we can show either an increase or decrease in binding. And in cases where we show an increase in binding over 200% of that wild type, we call that improved binding as well. So we're able to attune affinity to a target or off target with this method. And I have an actual sort of real life example of that in our hands. Again, another Claudin example where we were working on a Claudin 6 asset. This was, Claudin 6 is a tumor restricted protein. It's not really expressed endogenously in normal healthy tissues, but its family member Claudin 9 is. And it's very broadly expressed, and that can be a major risk to safety if you have any cross-reactivity there with Claudin 9. So as I mentioned, the Claudin family members are very similar to each other. These two, Claudin 6 and 9, only differ by 3 extracellular residues. And one thing, again, to go back to the whack-a-mole metaphor, one thing that is readily apparent to anybody that has done this engineering is that oftentimes when you tune affinity for one thing, the affinity for the other changes in parallel as well. And so if you gain affinity for the actual target, you can also usually gain affinity for the off-target in a lot of cases. So what we did here, this graph is essentially representing a single line from the previous table that I showed you with the blue and yellow and white boxes. And what I'm showing here is that, for example, in mutation A93Q, which is this bar here, we're showing great increases in affinity to the target, to Claudin 6, but the Claudin 9 off target also rises over 200% of that wild type as well. So again, we're dragging that up. Now, if we look at A93S, it's not the highest gain in affinity for the target, but what we're showing is over a 200% gain in affinity for the target with no change in affinity for the off target, for Claudin 9. And so what we're able to do here is we're able to tune affinities sort of independently of each other so that we can get the best possible sort of sequence that allows for that binding and function of the molecule in the most optimal way. So you can imagine sort of the implications here for also literally removing an off target. We can assess for mutations that have no binding to the off target, et cetera. So this is just kind of to show you that this is the way that the field is moving. It's the way that we're moving. And so engineering out these tricky off targets is kind of the next step after you've detected them. So hopefully you found that compelling. I just want to end again to say, end on this slide, and I think the next slide is my last slide, just to say that we do a lot of different things at Integral. I've spoken mainly about the membrane proteome array and the CDR scanning here. If there's any questions about these, I'm more than happy to take all of those. But if, again, you see anything on here that's interesting, we do have a division of our company that makes reagent monoclonals as well, some of the best on the market. They're all screened on the MPA for specificity. I'm happy to talk about anything else here as well. So with that, I wanted to wrap up and thank you all for your attention today. And I'm happy to open the floor to any questions that
Thank you so much, Alex, for sharing your incredible insights with us today. I know our audience has gained a lot from your expertise. Now, before we dive into the Q&A, just a quick reminder to our audience, if you have questions you'd like us to cover, go ahead and pop them into the chat box on the right-hand side of your screen. We'll do our best to get through as many questions as possible in the time we have. I can already see some really thoughtful questions coming in. Thank you for those. So let's jump right in. The first question being is, how do you ensure proteins are expressed?
Yeah, it's a great question, and it's one that we get quite a bit. I think ensuring protein expression is really important for just to speak to the quality of the assay. So the main way that we do that, the primary way that we do that is we have a V5 tag, an N-terminal V5 tag on virtually every protein in the library. There are some, one or two here or there that we weren't able to attach V5 tags for. But we can measure that V5 tag for expression. We also include a GFP transfection control, so we can ensure that transfection is occurring, but then also the protein is being expressed. And because we assess binding both intracellular and extracellular, we can actually get at that N-terminal V5 tag for all of the proteins, regardless of whether they have been fully trafficked to the cell surface or are somewhere else within the cell. So that's generally how we do it. We have a few more sort of more specific quality controls in place, but the V5 tag is how we ensure protein expression.
Thank you so much, Alex, for your thoughtful response. Moving on to the next question now. Is the initial MPA screening also performed on unfixed cells?
Yes. So just to clarify, all of the steps that we're performing are performed on unfixed cells. So we don't use any fixatives in our sort of binding conditions when we're adding that primary antibody to or your test article to the actual array. And so for each of the steps, the assay setup, the screen, and the validation, we are assessing binding in unfixed staining conditions.
Thank you so much, Alex. Moving on to the next question now. An audience member is asking if this research can be linked to study tissue investigations.
Yeah, I would say, the research regarding the link between MPA data and tissue investigations is really sort of, I would say, dependent on your specific needs as a company or as a researcher. I think I'm not, tissue engineering is not really my forte. It's not really my background, so I can't speak to that in particular. But I know that, especially in tissue engineering, there's a lot of omics work and a lot of generation of big data. And so the MPA could potentially be a part of that. I would need a little bit more information from the asker to get more specific than that, but that's kind of at a surface level.
Thank you so much for answering that question, Alex. Moving on to the next question now. On what stage should I screen my therapeutics for off-targets?
That's also a very good question. I think, again, it kind of depends company by company and program to program, but we always encourage people to screen early and screen often. That's kind of our motto. The earlier you can detect an off-target in drug development or antibody development, the earlier you can either address it or move on to a new lead. So that's why we really have kind of both options. We have the lead selection option for earlier stage candidates and the IND stage, the MPA plus IND service for later stage candidates. So early and often is our motto, but really you should be doing it at some point regardless. And I think one thing that we really prefer to do when we work closer with sort of true partners is really understand their drug development workflow and how we can fit in there in the most sort of efficient and productive way. So we can work with you to kind of determine when the best time is for you and your programs. But the earlier, the better, in our opinion.
Thank you so much for that thoughtful response once again, Alex. Moving on to the next question now. Once you've scanned all the single mutations, how do you know which ones to combine? Do you have to test every pairing?
Yeah, so I believe that's probably a question about Paratope Plus and the comprehensive mutational analysis. So the knowledge of which mutations to combine really comes from that sort of heuristic machine learning model that I believe I mentioned. And so what we can do is we can actually use our own data sets, and we can feed that to this model to understand better how to predict efficient combinations, essentially. And I know that the protein engineering team actually has very good sort of evidence and a good data set to show that we can actually improve things quite significantly with those mutations. We have good ways to analyze that. We do provide the ability to test a subset of those combinations. We can produce some of those for you. It does depend a little bit on project scope, but for anybody interested in that, I would say definitely talk to the protein engineering team, either through an introduction through myself or through our website or any other means, and they can give you the full scope of what that sort of combination testing looks like and how we derive those.
Thank you again for that response, Alex. Moving on to the next question now. Can CDR scanning be done faster or cheaper if a smaller subset of mutations were made and tested?
Generally, yes. I think the scope of these projects, again, this is a Paratope Plus question, the scope of these projects really depend on sort of the library size and what we need to generate for you. So there's a few different factors that go into that, and each project is going to be slightly different. I do know that if time and cost are important, which to many folks and many companies they certainly are, we can work with you, and we can work with you to design the best experiment possible for your aims. So again, kind of similar answer. If you have a specific question in mind or you have a specific project in mind, it really helps to consult with us, because we do a lot of sort of consultative back and forth with our customers to design the best experiments.
Thank you so much, Alex, once again for that most thoughtful response. I'm quite wary of time, so I'm going to ask the final question now. Do you make, express, and screen all of the single point mutation variants in the wet lab or CDR scanning, or is it done in silico?
Yeah, another great question. So kind of to clarify, similar to the unfixed cell question, all of the mutations that we're generating, all the data that we're generating for you in that primary data set is all done in our lab in Philadelphia. That is experimental data that we're generating for you. The only sort of in silico analysis portion comes with the combination analysis using the machine learning algorithms. So we are, as part of the power of this platform, is just the volume of data that we can generate. And there's a lot of implications there also for using that data in other AI applications, essentially, as well. So yeah, we're generating those large data sets experimentally in the wet lab.
Thank you so much, Alex. Unfortunately, that's all the time we have for today. For those of you who submitted questions that we didn't have time to address, we will be sure to send these over to our speaker after the event. Speaking of which, don't miss our next upcoming episode on therapeutic antibodies in transition, multi-mechanism strategies driving the next wave of innovation. A big thank you to you, Alex, for the insightful presentation and for addressing all of those questions. And thank you to all of you for joining us today. Before you head out, don't forget to explore the excellent supporting literature provided by our sponsor, Cynobiological, to further your learning. These resources are available in the handout section. We look forward to welcoming you back for the live for the next episode of our Therapeutic Antibody Series on 18th of August. Have a great day.
Thank you.